All right, everybody, we're going to get started. All right. Welcome, everyone, to the 2020 Austin San Antonio Vision Seminar. My name, oh, thank you. Little clap. Thank you. My name is Donna Cohen. I am the Director of Events for the West and Southwest region of the Foundation Fighting Blindness. I'm very happy to be here today. I have been with the foundation for over 13 years, and I'm very excited about the information that's going to be shared with you today. Before we get started, I just wanted to acknowledge our exhibitors that are here today in the room to my left or to all of your rights. AGTC, Austin Council of the Blind, Capital Metro, Eschenbach Optic of America, Inc., Open Eyes Life Coaching and Consulting, Spark Therapeutics, Talking Book Program, Texas State Library and Archives Commission, Texas School for the Blind and Visually Impaired, Texas Association for Parents of Children with Visual Impairment, Travis Association for the Blind, University of Incarnate Word Eye Institute, Vanda Pharmaceuticals, and VisionWorks. And please, when you have a moment, please make sure to go out and visit all of our exhibitors. They're very excited to be here and they would love to talk to you. There will be some time after the meeting and there will be a break. Now, just a couple of notes before we get started with the presentations. I just want to talk a little bit about today's agenda. We are going to have our welcome, and then from about 10:15 to about 11, we will have our panel discussion with Dr. Sarah Chexel, Jill Dolgen, and Chirag Javari. At 11 a.m., we'll talk about our upcoming Vision Walk that we're going to be doing here in Austin in May. And at 11:30, we're going to have talk a little bit about our Austin San Antonio chapter. We will take a break at 11:45. We will also have some snacks available, so there will be some different food available in the food room. And then at 12 o'clock, we will have some research updates with Ben Shaberman, who is from the Foundation Fighting Blindness. We will probably end at about 1:10, and the exhibitors will be available until 1:30 for you to visit and talk to them. I also wanted to just do a couple of housekeeping notes. For those of you, bathrooms are to your left. There's a hallway and a little bit of a kind of like a little path, and then you're going to make a right, and then you will be available to the bathrooms. Breakfast food will be available until 11 a.m., and then of course at 11:45 we will have snacks. Also, for all of you who have a cell phone, please put your cell phone on silent. And anybody who needs to hold a conversation, we ask that you do take that conversation out into the hallway so that those who are here can continue to hear what's happening on the stage. I also want to mention that the low vision aid store that is here will be open during this presentation and will stay open until 2:30 for anybody who would like to stop by and see if there's anything or an aid that you would like to look at or purchase. Also, as mentioned, our exhibitors will be open until 1:30. Please save all questions. And we also are recording this meeting on our Austin San Antonio Facebook page, which we can get that direct link for you. And I'm sorry, sir, and there was a question?
Where is that low vision aid?
So the entrance that you came through, it's right near the front entrance, and there will be volunteers there to direct you. All right, before we get started, I did want to thank all of our presenters, our panelists, our exhibitors, and the Chatlos Family Foundation for their support of our public health education program of the Foundation Fighting Blindness. I also want to mention in your bags, there are two pieces of paper in there that I would like to bring to your attention. One is our raffle card that you can fill out, and another one is an event evaluation, and the event evaluation is anonymous. We ask that you fill both of them out. First of all, the event evaluation to let us know how we did, how we're doing, and what we can do better in the future. And also the raffle card is for anybody to talk about how you might want to get involved. And we're also going to use that to give away a couple of prizes. And volunteers will be coming through the room to collect them or to assist anybody who needs assistance filling that out. And we'll remind you as well. All right, at this time, I would like to start our panel discussion with Q&A. Connor, do we have someone that can manage this? Bear with us for a second. Okay, and it's on. All right, so our panelists again are Dr. Sarah Chexel, Jill Dolgen, and Chirag Javari. Dr. Chexel and Dr. Javari are with Retina Consultants of Austin, and Jill Dolgen is with AGTC. So let's welcome them as our panelists. I'm going to have to stand, sorry. All right. So first, if we could start, if each one of you could please give your name, position title, school of organization affiliation, and your particular area of expertise in retina.
My name is Chirag Javari, and I'm with Retina Consultants of Boston, as well as the Retina Research Center, and a clinical assistant faculty at the Dell Medical School. We have a research center that we focus on with clinical trials and clinical studies for patients with many retinal conditions, including macular degeneration, as well as diabetic retinopathy, as well as other vascular conditions that can affect the retina, as well as potential surgical studies for delivery of medications to the back of the eye.
My name is Sarah Chexel, and I work with Dr. Javari at the Retina Consultants of Austin, as well as the Retina Research Center. We work together on the clinical trials as well.
Hi, I'm Jill Dolgen with AGTC, which stands for Applied Genetic Technologies Corporation. We're a biotech company based in Florida and Boston. We're an early stage development of gene therapies for patients with X-linked retinitis pigmentosa, achromatopsia, and some other products in development. In terms of my background, I'm not an ophthalmologist, although I play one at work. I am a pharmacist by training, and my job at AGTC is really about education, to both for patients, healthcare professionals, caregivers, and my one goal is really to empower you with the knowledge you need to take an active role in your care, and whether that means getting into a clinical trial or a registry, but more importantly, finding out more about your condition and what genetic mutation you may have. I think that's the first step in understanding what those options might be. So my goal is really about education.
Thank you. What research and/or clinical trials have you most recently been a part of? What is the current status of that, and how do you feel it will influence the field of inherited retinal disease?
Do you want me to start? Sure. So X-linked retinitis pigmentosa, you may be aware that retinitis pigmentosa is a big disease. There's probably over 200,000 people in the US and Europe that have retinitis pigmentosa. And there's over 200 different mutations that can cause that condition. We're actually focused on about 10% of those patients that have the X-linked form, meaning that it was passed down through the sex chromosome, through the X chromosome. So that means that the females are carriers. And we've just finished really early stage, meaning phase 1, phase 2. And the goal of phase 1 and phase 2 trials are to determine whether it's safe, number 1, and number 2, whether there's some evidence of biologic activity. So we have just finished enrollment with X-linked RP. And the goal now is to go to the FDA, have a conversation about the results of that trial, and figure out whether we can move forward with a phase 3. In phase 3 is where we would enroll larger population, probably 45 to 50 patients, and treat both eyes. So in phase 1/2, where we're trying to figure out, is it safe? We're only treating one eye because we don't want to do any damage. We would just want to figure out, is this gene therapy safe? But in phase 3, where we've determined what that highest, safest dose is that shows some evidence of activity or efficacy, we now want to treat a larger number and see if this is something that we can bring to market for a much larger population with X-linked RP. Our achromatopsia trials, and achromatopsia is also a rare genetic disease of the eyes, where individuals, it's primarily a cone disease. So that means that your patients have severe light sensitivity. They have poor visual acuity. So your cones are responsible for your fine vision, your light vision in the day, and also responsible for color perception. And so those with achromatopsia, of which there's maybe 20,000 of those in the United States, there's 6 different mutations that can cause that condition. So, but we're focusing on 2 different mutations that account for about 80% of those. So we're just about finished. We're finishing up enrollment of those 2 trials. And again, over the next year or so, we'll be able to determine whether the data looks good enough for us to talk to the FDA and go on for a phase 3. So we also have a number of rare genetic eye diseases in our portfolio that's still in animal studies. So Stargardt's and AMD, very early, probably won't be in the clinic for a clinical trial for probably 2 to 3 years at least.
So I think this is a really exciting time for the community and patients as a whole who have inherited retinal disease. I sort of think of retinal clinical trials divide them into 3 large groups, categories. The first one is gene therapy, of which you heard a little bit about that already. And there are several other companies that are also working on gene therapy for their own specific genetic mutation that they might be interested in. As you all probably have already heard, Luxturna was the first FDA-approved gene therapy treatment for retinitis pigmentosa and a specific gene called RPE65. And so the challenge with gene therapy is it's sort of like trying to make it a plug-and-play reusable model. And there's certain genes that are amenable to gene therapy due to size, and other genes that are not due to being very large in size. In gene therapy, the way that it works is you basically have a virus that carries the good copy of a gene into your DNA, and then it incorporates that into your DNA and essentially rewrites and overrides the faulty copy. And as you can imagine, that's fraught with complications depending on the size of the gene. So that's one category, and there's several companies that are involved in gene therapy trials for that. NIBR, which is a Novartis company, is looking at gene therapy for one specific genetic mutation called RLB1 mutation, and this trial is being conducted out of Sweden at the moment. The second category that I like to think of clinical trials for RP is stem cells, and you all have probably heard a lot about stem cell trials and what that means. But the benefit to stem cell therapy, if it works, is that it's sort of agnostic to your genotype, meaning that it can work for any genetic mutation for any cause. Because essentially what you're doing is you're reprogramming the stem cells to remake copies and proteins and structures within the eye that are perhaps faulty. And my partner will talk a little bit more about stem cell trials and the pitfalls of that and what to look out for when you're talking about that. And then you all have probably heard of CRISPR and gene editing, and that's the other sort of newer frontier in clinical trials. And that's sort of where you, rather than a virus carrying a good copy of a gene into your DNA, you essentially have molecular scissors, if you will, that sort of just cut and remove the faulty gene and then replace it with a good copy. And that's interesting in its own right because it's sort of independent of the size. So it's able to work through some of the challenges that gene therapy so far has not been able to. But CRISPR and gene editing is still in the early phases. We still don't have data to show safety in humans as of yet, although it's very promising in mice. There is a gene editing clinical trial that's being conducted out of Columbia University at the moment, where they're looking at autosomal dominant retinitis pigmentosa, which is actually sort of the one area of RP that has not yet been really studied too well in terms of treatment. And that's because it's incredibly difficult to treat two faulty copies of a mutation. It's easy for autosomal recessive or X-linked disease to replace the one faulty copy, but it's incredibly challenging to identify both faulty copies and replace those without harming anything nearby that might be a normal, good functioning gene. So that's sort of where we stand, and we've made a lot of progress in the last 10 years, and we've got a long way to go, but it's exciting times.
That was very well said, and I agree. I think these are the three large areas that we can look at for long-term treatment. The problem with that is that it is very, very challenging to have those types of cures and treatments because we're fundamentally changing or trying to change either the genetics of somebody or trying to regenerate or regrow structures that have been severely damaged. And so, the other thing that I also think about, and this is in some ways almost maybe a stopgap, but we are probably potentially closer to something than a cure-all for many of these inherited retinal diseases, is bypassing some of the visual systems that are damaged in order to try to improve vision and try to improve functional vision. So for example, camera systems and implants that will bypass the optic nerve in order to allow patients with very severe vision loss to have some functional vision, albeit a very crude way, almost like a very kind of low resolution pixels, as well as using implants underneath the retina that bypasses the damage from retinitis pigmentosa, for example, in order to transmit once again a crude picture that gets sent to the optic nerve. So the Argus 2 prosthesis is an example, but there's several others that are being developed around the world, including Australia as well as England, that are being used for that. And I just want to touch up on stem cells. We have Dr. Golden, who's part of a genetics company, which has a wealth of knowledge regarding that. And I think that that's probably going to be one of the better ways to try to cure this. But as Dr. Chexel mentioned, it's very hard. Everybody's, retinitis pigmentosa has more than 200 different genes. So even if you develop a method of treating one patient, one type of mutation, ideally, if you can replicate that, you still have to replicate that 200 different times and find and verify that process for every single mutation. So it's very challenging. Stem cell seems and has been in the news a lot, and it seems much more promising. There's magical, wonderful cells that can turn into parts of your bodies or tissues that are damaged and will regrow. And it seems amazing, and it is amazing. And I think there's a lot of potential for that. But it is still very, very complex. And when people hear about stem cells and injecting them into knees and whatnot, that's very different than doing that for the eye. I would, think about the eye as a very, very complex organ with very many different parts that are completely unique and different. Similar to like a very complex pocket watch, where you have a lot of different gears that are of different sizes that do a lot of different things. So when you have something like stem cells that has the ability and potential to turn into many different things, you want to make sure that it's guided properly. And that's some of the dangers and difficulties for stem cells for ocular conditions. The eye is made up of so many different specialized cells that there's a chance that if it's not done properly, it will turn into random tissue that's not meant to be where it's at. And an example, as opposed to somebody who's getting stem cells in the joint and you just want cushioning, you just need some extra tissue to be made, you just want some extra cushioning to be made, and obviously maybe a certain type, but it's less important about if it's done and made in a very specific way. So stem cells, I think, are promising, but we have a lot to go forward to go on in terms of trying to make sure that they're done. I think, in a simple way to think about it is I think we need to make sure that the instructions that are then brought, or we have instructions that are delivered with stem cells so that the stem cells can turn into the right type of tissue in the right shape, in the right manner. So possibly like a scaffold that's delivered with the stem cells and whatnot. The other thing, of course, I do, and I think some of the questions that we're going to talk about, but I want to touch briefly is participating in clinical trials, and to be very careful. Clinical trials are carried out often with conjunctions with researchers, investigators, funding both publicly and privately, and often in a company as well who has maybe access to a technology. And they're all registered on clinicaltrials.gov. But I also want to let you know that clinicaltrials.gov does not verify what these clinical trials are specifically about. So there's a few things that I would recommend and pitfalls. And the reason why is because I wanted to bring up how there are, advances in stem cells that were reported several years ago. And if you read the details carefully, some of these clinical trials are done where the patients have to pay for the treatment themselves. And I would be very wary of things like that. There was a very famous situation in Florida where there were reports of stem cells being injected and some patients doing very well. Well, unfortunately, there were also many patients who lost even more vision because of the complications that occurred. And these were patients who had to pay for their treatments. And typically, research is not conducted in that way where you're having to pay for an experimental treatment. And so I would be very wary of something like that. And those sometimes can be, unfortunately, a little bit more shady situations. And the reason why they were able to get away with it is because of loopholes and technicalities of the laws. Because many of times these stem cells and, there's autologous stem cells that are basically taken from your own body versus having stem cells from a line that's derived from a company or a university having those stem cells from somebody else. So these autologous stem cells, because it's in your own body, it's kind of like there's nothing new or different. We're not delivering a new medicine to you. We're not delivering a new device to you. And so it basically escapes some of the rules and regulations that are usually set forth for performing clinical trials for new treatments. And so that's why there's this pocket where you may be exposed to that. So I would just be wary of that. I always recommend people, if there's ever a question or a concern, to get a second, if not even a third opinion about things. One of the things always is if there are multiple sites, if there are, multiple people involved in this, that always can be reassuring. But still, it can be very difficult to make sure that you are understanding the situation. But if there's anything a little bit unusual about it, I would just make sure that you do a little bit more research and digging.
Thank you. So we were talking about clinical trials, and we know that it is an arduous process. How would you address an individual that has been told there's hope, but does not feel they have seen the evidence of that in current treatments?
Yeah, I'll just talk really briefly, and then I'll have Dr. Chexel also talk a lot about that, I think. But, in general, research is a very, very long process, particularly with very rare conditions. It's hard with rare conditions, there's less people that we have with less data. And some of these rare conditions, particularly that we've known about for 40, 50, 60, 100 years, are also changing because of our understanding of genetics, actually. So many times these conditions that were we thought were to be one single type of disease, we find out are maybe 5 or 10 or 15. And so therefore, it's a very long process, and rightfully so. It also takes a long time to make sure that something is safe and effective. So many times with these rare conditions, we're starting just at the beginning to try to even understand if the way we're doing something is the right starting point, let alone step 3, 4, 5, 6, or 10. So even when something is promising, that promise could be often a decade plus of time that we need to spend.
And I think that we always tell our patients to remain hopeful. And the reality is that's because of two reasons. Number one, we're doing a lot of work in clinical trials. We're making advancements. Just a few years ago, gene editing wasn't even possible, and now it is. So things can rapidly change in the space. But the other thing is we're becoming more educated and more informed. Just a few years ago, we only knew of 129 genes that were the causative mutations in retinitis pigmentosa. That number has increased to more than 200 now. And that's just in a few years. So now we're becoming better at identifying the cause of these, the pathology. And so that's really, really important because we need to know what we're treating before we treat it, number one. And then number two, our technology is advancing at such a rapid pace that we're actually able to make huge strides. And I think gene editing is one of those areas that's super promising because of that. As we identify more and more mutations, we can quickly go in and splice those, cut those mutations out and replace them. And I think, and so we always tell our patients, remain hopeful. Obviously, science is a process. It's a journey, and it takes decades to do good work. But we're also at a time, we're sort of at a precipice where technology has advanced so rapidly, and our knowledge and understanding of the disease is advancing at such a rapid pace that they're catching up to each other, and we're able to do more and offer more in a shorter period of time.
Thank you. I'd like to make a couple comments. I go around the country and I talk to patients, I talk to families, I talk to healthcare professionals, and they, to your point of, well, I'm not sure I'm ready to join a clinical trial, what can I do? And as a patient advocate, I tell you there's 4 things you can do that are really going to help us advance the science. Clinical trials are one of them, but that may not be right for you, and there may not be a clinical trial for you. But there's things that you can do. One is keep telling your story, because everyone has a different progression of their disease. So even if you and your family member have the same gene mutation, the way your condition progresses is going to be different. So when you go to one of these ophthalmologists, you're going to tell your story and they go, well, you've got the same mutation, but your vision is very different. So we're learning from that to better understand the disease. The second thing, and probably more important, is we're learning more and more, because now we have all these gene mutations that have been discovered. They've only been discovered because we've been doing more testing, right? So the first thing you can do is to help us advance the science. Clinical trials is almost the last thing, is to get genetic testing done. And there's free genetic testing available now through Foundation Fighting Blindness and a couple of other companies. So find out what that mutation is, and then contribute to the science by joining a patient registry. So the Foundation Fighting Blindness has over 25,000 people in that registry. So we as industry specialists, when we decide what we're going to do next and what target we're going to shoot for, we say, well, how many patients are there? Where are they? These are rare diseases. We're going to go to Foundation Fighting Blindness and say, out of those 25,000 people that are in your registry, can you tell me what numbers, how many of those with XYZ disease do you have? Because that's going to help us determine whether there's an unmet need out there, but it's also going to help us understand the genetics. So there may be 500 of those individuals that have that disease that we're considering a therapy for, but every one of those 500 have a little bit different mutation, right? And so we need to understand that when we do administer a treatment, we need to figure out, of those with X-linked RP, for example, and as we're looking at our data, is there a subset of those X-linked RP patients that are doing better than the others. One, because of their extent or severity of their disease, but we're also looking at, well, where is the mutation in that gene? Okay, it's like a long sentence. Where's the word in that sentence that actually has that mutation, that has that damage? And is there a commonality? So if, 10 of your 20 patients respond to that gene therapy, is it because all 10 of them have that broken word, that mutation in that same spot within that gene? The only way we know that is by you contributing your information to a registry. And also the last thing is to join a natural history study. So a natural history study is a non-interventional. It's basically conducted like a clinical trial, where you go to a clinical trial site every few months for maybe a couple years, and they're doing all these tests and collecting that in a systematic way, so that we know if you come in and get treated and we give you a treatment and we say, have we done anything to change the course of that disease? The only way we know that is because you've now part of a natural history and we've seen how your disease has progressed over that previous 2 years. Now we can see moving forward, have we slowed it down? Have we stopped it? Have we improved it? So clinical trials are great. There may or may not be one for you, but joining a natural history study and joining a registry are two things that you can do to help advance the science. The other advantage to you, aside from contributing to science, is that you're going to be in line to be communicated with through a letter from the FFB or other ways to say, because you've been in this natural history study, because you're in this registry, you've been identified, we know you have this condition, and guess what? This pharmaceutical company or this biotech company is now conducting a clinical trial. Thanks to you, we know there's an unmet need and there's a large population out there who may qualify for our trial. So putting a plug in for FFB, but more importantly, it's what I believe as a patient advocate and when I go around the country talking about. So thank you very much for that question.
I completely agree. And also, if you have not been genetically tested yet, there are free ways, like as was mentioned. Usually, any retina specialist, particularly if they're doing any type of research, will have an easy access to that. The FFB can also help provide that. So if you are seeing a retina specialist, ask them. For example, we have kits already in all of our clinics that can test for that, and it is free. Currently, the kits that are currently available typically will test for the majority or the most common types of diseases. If you happen to have a vision-threatening or inherited disease that does not match that, then the next step can be a little bit more comprehensive genetic testing. Those sometimes do cost some money. Either sometimes insurances will cover it, but sometimes they won't. But fortunately, because of our database as it's growing, we're able to kind of identify the most likely causes with up to 250 genes. So that's a lot of genes that we can test for a lot of different varied pathology of diseases. So I want to echo that. Please, if you haven't already, get genetic testing if you've been diagnosed with an inheritable retinal disease, because now it's free.
So on the subject of genetic testing, which we will talk a little bit more later on in the afternoon as well, how would you advise for those, one, who have had their gene identified, and two, for those who have been tested with no result? What would be your advice to those patients?
So for those who were genetically tested with no results, there's a couple of different ways that we can approach that. The first is that it's not that they have no result. It's just that we may not have identified which mutation is the pathologic mutation. In other words, sometimes when we get genetic testing, we receive a report back that says we received 4 or 5 variants of unknown or undetermined significance. And that just means that we just don't have enough information about whether or not these are actually pathologic mutations. So, and perhaps they are in that particular patient. We just don't have enough of a normative database to compare them to say, okay, this must be the mutation that is the causative. If you've been genetically tested and you have a mutation that's been identified, I agree. I think that it's really important to get registered through FFP, or there's a Identify My ID My IRD website as well. That's really beneficial. And I think it does that for a couple of reasons. Number one, it gives pharmaceutical companies, biotech companies, a good read on how many patients are actually have that mutation, because that helps obviously with selection of what their next target is going to be. But it also can put you in touch with other similar genotypic patients, and that could be helpful because we know that that the genotype does play a role in the rate at which your disease progresses. And within that, of course, there's a spectrum, and certain people who have the same autosomal dominant mutation might progress at a slower or faster rate. But it's helpful because there's certain mutations that we know progress much faster than others, and certain mutations spread in a very particular and peculiar fashion. And so it'd be helpful to just sort of be able to contact others that have the same mutation as you and support each other in that way.
Also, just quickly to add that a lot of times they'll get updated. So what will happen is that you'll get you'll get your report back and it'll say of uncertain significance. And there's a chance that because we're getting more and more data, there's more trials, there's more basic science research also being done, that it will then update and say that we've had several patients who've had that, where I've then been sent an updated report about their genetic testing. So so just because when you first get your report, we don't know, doesn't mean that it'll always be that we don't know.
I would just say the genetic mutations are like a library. It's a database of information, and it could be that 10 years ago when you had it done, nobody ever reported that mutation had before. And now 10 years later, we've now got hundreds of people that have now had testing and they have that same mutation. So the database is changing all the time, but that's only one piece of the puzzle. So the doctors are going to look at you and say, well, it says unknown significance, but I look at the symptoms that you have and the parameters of your eye disease, and it is seem to be contributing to it. There's nothing else in this gene profile that suggests that that could be causative. So even though the lab result says it's unknown significance, the doctors are going to be able to look at that information, look at your eye condition and understand now, that's another piece of the puzzle to help us understand whether that's causing your condition or not. So it's not just one piece of information, it's along with everything else that you're presenting with.
So before we open it up to Q&A, I wanted to ask one last question of each one of you. What do you feel the greatest promise for treatments of the retina for those with, what do you feel are the greatest treatments for the retina for those with an inherited retinal disease? Like, what do you think holds the most promise?
Well, obviously we believe ours hold the most promise, but as a patient advocate, I always ask what's next, right? Is there a way that we can combine modalities? So for example, we talked about stem cells, we talked about gene editing. Is there opportunity, and I know that there's, Ben Shaberman may talk about this, is there a way to combine them? Are we doing studies now in mice or other animal models that allow an individual to receive more than one type of gene therapy at the same time or sequentially? So maybe you get gene therapy now, and in a year after that, you get a stem cell treatment, or can we combine it into one type? So I think all of these modalities hold a lot of promise, but you have to understand that research is an arduous long-term process, and it takes 15 plus years to bring something like that to market. And in the early stages, phase 1, 2 that we're in, there's a 90% failure rate traditionally in the pharmaceutical industry, 90% failure in the early stages. And so even going on to phase 3, there's no guarantee that it will eventually show enough efficacy that we can bring it to market. So there's a lot of promise, but it does take a long time, and we depend on individuals like yourself. I'm always in awe when I talk to individuals and they say, I know it may not help me, but I want to contribute. And I'm speaking to children and adolescents who say, I know it may not help me, it may help me, but in my lifetime, I want to give back, I want to contribute. So adults, kids, it's a, only because of people like you are we going to be able to advance the science to a point where we can hopefully bring a cure to market in the near future. Thank you.
Yep. So I think that's a great point about merging technologies. And I think, fortunately, we're at a place right now where we don't have to pick one. We don't have to pick only gene therapy to advance or gene editing or stem cells. We can sort of merge the technologies. And there is one researcher in New York who is merging gene editing and gene therapy at the moment. And that's very exciting because if you can cut and remove a mutation and then you can follow that up with a adenoviral associated vector that carries the normal copy into the cell, then maybe we've hit a home run. So I don't think that it's about picking which one technology is the best. They're all great. And the thing is, they all need a lot more work, but there are researchers that are willing to do that. There are pharmaceutical companies that are now willing to take that on, biotech companies that are now interested in this space, whereas just a few decades ago, people were not, biotech companies were sort of going after more low-hanging fruit like wet macular degeneration. But now we have a real interest in this, and I think that is a testament to all of you. I think if you advocate for yourself and you make the community aware of what you go through, then it really brings that to the attention of biotech companies.
So I think ideally, in its best way, I think I agree, gene therapy, either if it's editing or transfection, is going to be the best way. A way we can think about some of this is depends on where you are, right? So gene therapy is almost like a vaccine. And ideally, in its most ideal way, we identify somebody has that problem before there's a problem. And when we give them that therapy before any damage develops. Things such as stem cells are almost like antibiotics, where there's now damage that's being done and we have to try to get rid of that. So, in many ways, for example, many of our current patients who have already lost a lot of vision, gene editing may help them, possibly prevent them from losing any more vision, but it's unlikely to regain any of their vision. So that's where stem cell comes in and where we can maybe, replenish and regenerate, as well as maybe, implants that can bypass damaged pathways currently. In the most ideal setting, we won't need those other than maybe very few situations and rare situations where either somebody was missed or somebody had trauma or something else. And ideally, we have gene therapies that when you're born or within the first few years of your life, those are identified, we can treat that and you never have a problem and you're living a normal life moving forward. And that's really, I think, the way I see it, the ultimate goal. Is that near? Not for a lot of conditions, no, but we're starting at, as we have the first gene therapy available for a very specific type of mutation. And as we're continuing to look for other very specific mutations, but that's where we're at. So all of them are exciting. I think in many ways, as we develop these will be merged together with eventually us having kind of almost a preventative means where no one ever has to go through the process of losing their vision.
Thank you. All right, now we would like to open it up for questions. Please raise your hands, and Connor here will come to you with a microphone. Connor,
We're going to take this one question at a time, so please make sure that you are raising your hand. I only have one microphone to go around.
Is there anything that you can take as far as supplements or whatever to keep your condition from getting worse? Or vitamins or anything?
Yeah, there are a number of studies that look at vitamin supplementation in patients with RP specifically. The original paper talking about vitamin A supplementation was written by Dr. Burson years ago. And what he found was supplementing your diet with 15,000 international units of vitamin a day was helpful for some patients with retinitis pigmentosa. However, that paper and that advice was sort of over extrapolated over the course of many years. And what we now know is that vitamin A has a lot of toxic byproducts. Vitamin A is not just a vitamin, it actually has retinol, retinoic acid, retinal, and some of those are very toxic to certain types of genetic mutations within RP, specifically like ABCA4 and others. And some young children with RP are very susceptible to high levels of vitamin A. So what we recommend is that if you do want to start on vitamin therapy, vitamin A specifically therapy, talk to your retina specialist about it first. See if that's something that they would recommend that you do given your genotype or your age or the type of mutation that you have. Secondly, there's some talk about lutein. Lutein is found in it's a carotenoid and it's found in macular pigment. So it helps protect the fovea from some of the shorter wavelengths of light. Lutein is found in, it's found in a lot of vitamin supplements that you might already be taking. So I don't think you need to supplement on top of that. But if you don't have a vitamin that already has lutein in it, maybe consider switching to one. And then there are other antioxidants that you can also take that have been shown anecdotally to help with protection of the eye pigment.
So,
the antioxidant DHA, for instance, has been shown to, again, anecdotally, not in large-scale clinical trials. So, but once again, these are antioxidants and vitamin supplements that usually are not toxic. Vitamin A is the one exception that you really do want to talk to your retina specialist about before supplementing high doses of vitamin A. But the remainder, are found in most vitamins that you probably are already taking, multivitamins that you're taking over the counter.
So supplements are, of course, it gives us potential hope that, maybe we can slow our disease down. And that's something that I do want to stress is that it is not a cure. If it was, we have looked at many of those already. So I think it's important to incorporate and maximize supplementation as appropriate. But you also want to recognize that, the genetics are the most powerful thing. So it's almost like, trying to, put some slightly broken brakes on a car that's running downhill. So it's important, but it's not going to be something typically, particularly for inherited retinal disease, that will prevent you from ever having a problem. Your genetics is most likely going to be playing the largest role. But as a corollary to your question about what you can do, like supplementation, I also want to stress what you shouldn't do. So, and that's certain things like smoking. Smoking is something that you don't want to do if you have an inherited retinal disease. That will further stress your retina, your optic nerve, and your vasculature. And if you already have a very delicate visual system, this will be putting a significant risk, a significant strain on that, and most likely will also contribute or put you at risk for additional causes, additional problems with your vision, not only with the genetic condition that you have, but in addition may also exacerbate other things. Overall, healthy lifestyles is always important. So no smoking, trying to make sure that if you have high blood pressure, that's under control, trying to make sure that if you have diabetes, that's under control, trying to make sure your cholesterol is under control. All of these are important. And so, for us, I think those are as important, if not more important than trying to find that specific vitamin. Because if you are living a healthy lifestyle and you're eating fruits and vegetables and green leafy vegetables, a lot of these supplements, in the fruits and vegetables, they're in there, and they're in at pretty good doses also. And as Dr. Chexel noted, a lot of them are not clinically proven in a large trial. So taking very, very high doses of something without us knowing for a fact, doesn't necessarily mean that it's going to be completely safe. And so you want to be a little bit wary of that as well. But usually the things that are given, if you're eating, generally healthy diet, those are not going to be at very high or toxic levels.
Okay, and then we have a question over there.
Before I get to my question, I just want to add to that nutritional discussion you were just having. I have Usher syndrome, and I have a brother that's in his 70s and also has the same genetic disorder I do. He's in his 70s and he still has functional vision, better than mine, in fact. But what he does is he walks a total of about 6 or 8 miles a day. He eats fruits and nuts. He's a nutritional freak. He eats nuts, seeds, fruits, vegetables every 2 hours. He eats just avocados, things like that. But he's gone away from meat, and he attributes his being able to still see into his 70s due to his rigorous nutrition and cardiovascular activity. But I guess my question that I had was, when we have these retinal degenerative disorders and sensitivity to light, when we go to see an ophthalmologist and they want to dilate our pupils, of course, and look into our retina, is there damage that occurs to our retina when you have these intensive lights into your retina so that they can see how it's progressed? Should we be concerned about getting our pupils dilated and undergoing these examinations?
That's a great question. So, we do worry about intense phototoxicity for patients with retinitis pigmentosa. In general, the dilation themselves should not be a significant issue. We have ways to prevent, even patients who don't have retinitis pigmentosa are going to be sensitive to light during dilation. So we usually have patients who are in mostly a dark room. Of course, you also wear sunglasses thereafter until your dilation improves. And the exam itself, oftentimes, the first time that you're seeing a retina specialist, there may be a longer visit because we want to confirm the diagnosis and do a good exam. And of course, we continue to do a good exam. But I will say that, anecdotally, when I do have a retinitis pigmentosa patient, I try to make sure that it's efficient. I'm not sitting around looking for, for a minute or 2 minutes. I'm not having a student, we have residents and students who will come through. I'm not having them, try to look at the retina and spend 5 minutes on those patients. I think the idea is to make sure that you have a retina specialist who's, you have a relationship with it and is efficient. And most retina specialists who see patients like that are. I don't think it's something that is of a major concern. The amount of light intensity that's there is not so intense that it would cause any significant change. But having said that, I don't have those patients be the one that as a teaching case or whatnot. If I have images, I show my students the images and use that as the teaching case often. So I think I don't want you to be concerned or worried in that way. Also, typically speaking, unless there's an active issue, we're usually doing these exams, either or most often about once a year. And those are, I think, important to make sure that there aren't other changes occurring. For example, cataract formation. Patients with retinitis pigmentosa can also develop swelling of the retina temporarily, and we want to look out for that and monitor that as well. And with our ability to image things, usually our need to have much longer exams with the bright lights has been fortunately minimized.
And I just want to add to that. I think there's been a lot of talk about blue light and blue light toxicity to the retina, and that sort of scare happened because of one particular physician investigator at the University of Toledo who exposed mice, not humans, to unnatural super thresholds of blue light. And these were mice whose eyelids were taped open and subject to 48 hours of continuous blue light exposure. And at that level, those mice experienced phototoxicity to the retina. However, we're all fortunately born with eyelids, and we don't have them taped open for 48 hours. And so even if you were to spend 42 days outside in the sun, you just can't be exposed to enough blue light that would actually cause damage to the retina. That said, I think for comfort and for other reasons, we often, and for contrast sensitivity purposes, we often recommend that our patients with retinitis pigmentosa, as well as other inherited retinal diseases and even macular degeneration, consider wearing amber tinted glasses or orange, blue, we call blue blockers, just to help with transitional lighting. And it does filter out some of those shorter wavelengths of light.
All right, we have time for one last quick question.
Hi, everybody. Good morning. My name is Zubian Wuta. Thanks so much for the FFB for organizing this. I have to give it a little bit of context before I ask my question. 96, my visual acuity was 20/40. 98, it was 2200. I was diagnosed with Stargardt's. However, my Stargardt's never stopped the visual decline. It never followed the classic symptoms. From 98 to 2012, I went through 300 eye care professionals, conducted my own genetic testing to find out that I have 3 defects to the ABCA4 gene. I believe that Stargardt itself has 3 kinds, and cone rod dystrophy, has its own problem. So the question I have is, how can we start distinguishing between somebody who has one eye disorder versus somebody who has multiple? Because if you look at my diagnosis, it says Stargardt's, and if somebody were to Google Stargardt's, it will say 2200. Use low vision magnifiers, you're fixed, you're fine. However, in my situation, 1 + 1 + 1 was never equal to 3. Thanks.
Yeah, that's a great question. I think that what you've hit the nail on the head here is that a genetic mutation, a genotype does not equal phenotype. So what your genetic mutation is does not mean that that's how your disease is going to necessarily progress. And yes, classic textbook Stargardt's, we all know, patients present in their first decade of life, their vision typically does not decline beyond 2200. But there's a myriad of patients that fall in between that spectrum. There are some that see better than that, and there's some that see worse than that. And the reality is it depends on how and where your Stargardt's starts and where it progresses to. So in the retina, it's really all about real estate. Where the atrophy in Stargardt starts, if it starts very close to your foveal center, the fovea is the centermost part of the macula that is responsible for your 20/20 vision. If that is affected first, it typically Stargardt spreads outward from there. And if that center area, the most critical and sensitive area is affected first, then yes, you would likely be the patient that would end up with worse than 2200 vision. So I think that the bottom line is that you're right. Having a patient with a genetic mutation, whether it's ABCA4 or ELOVL, which is typically the more aggressive genotype, does not necessarily portend what your phenotypic expression, what you're going to experience as a patient. And that's just because we're still learning. There's many different epigenetic phenomenon that occur. So it's not just the genetic mutation. On top of that, there are epigenetic mutations and other things that play a role in how this gene is expressed for you as a patient.
All right. Well, let's please give our panel a round of applause. Again, I want to thank all 3 of you for coming here this morning to be here. We will also, again, later in the morning, we will have an additional research update with additional Q&A period. So if you didn't get your questions answered during this period, please save them for the next period. Now, I would like to turn this over to my colleague, Michelle Goldfarb Shapiro, who's going to talk a little bit about what we're doing locally here in the Austin and San Antonio area. So Michelle, would you please join me up on the stage? Thank you so much.
Thank you, Donna. Good morning, Austin. It's so nice to be here with everyone. So I believe Donna mentioned this earlier, but everyone should now have a half-page interest sheet from us. And if anyone needs help filling it out, please don't hesitate to reach out to any of the Lighthouse or our staff members, and we'd be happy to help you. There will be a raffle at the end of the day, and there will be 6 prizes that will be awarded for people based on if they filled out their half-page sheet. So, I'm going to give everyone a little background on the foundation. For more than 50 years, Foundation Fighting Blindness has been dedicated to the discovery of preventions, treatments, and cures to restore vision and reverse blindness caused by genetic retinal diseases. Since its inception in 1971, the Foundation Fighting Blindness has pioneered leading-edge eye disease research with the passion, commitment, and funding necessary to launch an entirely new field of scientific inquiry. The foundation has raised more than $760 million. We decided from the beginning that we couldn't leave any stones unturned, no matter what, no matter how long it takes. The time is now to end blindness. Since the beginning of this year, retinal diseases will have claimed the sight of 50 million people across the globe and caused 100 million people to be visually impaired, most of them legally blind. We are now in 35 human clinical trials, but there is still so much work to be done. The foundation is committed to ensuring as many clinical trials targeting the broadest possible range of retinal diseases get underway before the end of this decade. I am very excited to announce that the Vision Walk has just crossed the $55 million threshold since we've started. So, this is our first year that we will be having the Austin Walk. In the past years, it's been in San Antonio. The Austin folks have had to drive to San Antonio to participate, and now it's time for the San Antonio folks to drive to Austin for the Vision Walk. Austin is a growing and bustling city with many wonderful resources for the low vision and no vision communities, and we are absolutely thrilled and honored to be able to have the walk in Austin in 2020. The 12th annual Austin San Antonio Vision Walk will be taking place on Saturday, May 2nd, 2020 at Mueller Lake Park and Hangar in Austin, Texas. Registration will begin starting at 9 in the morning, and the welcoming presentation will begin at 10:20. The ribbon cutting and walk will start at 10:20 a.m. What is Vision Walk? There is a photo on the PowerPoint screen against the wall that is of a woman cutting the ribbon at the entrance to our Vision Walk last year that took place in San Antonio. The Vision Walk is our signature fundraising event, and it is an unofficial 5K, we call it. So it's 3.1 miles, but it's not timed for any official race. There are over 40 Vision Walks held annually across the country, and it is a very family-friendly celebration. At Austin Mueller Park and Hangar, we are actually able to bring our dogs on leashes as long as they are on leashes, and we are more than happy to have pets there along with service animals. I was just given notification of this change. The 12th annual Vision Walk has occurred in the San Antonio region since 2009, and since then, it has raised over $1.2 million. Welcome to the $1 Million Raise Club. That's an absolutely fantastic accomplishment, and I just want to congratulate everyone who has participated in the past. So let's talk goals. This year's Austin San Antonio goal is $75,000. In this slide, there is a photo of a group of individuals from last year's Vision Walk posing for a photo in their Vision Walk t-shirts holding pom-poms. So at this time, I'd like to introduce our Austin Walk Chair, Miss Cece Collins, to the stage. Her team name is RP Visionaires, and I'd also like to just share the San Antonio co-chairs, walk chairs, are Todd and Meredith Dunn, and their team name is I Believe, and they will be joining us later to say a few words. So this time, let's welcome Cece to the stage.
Thank you, Michelle. Appreciate that. Welcome, everybody. Good to see everybody here. I'm just going to give a little bit of information about why we participate in the walk. I'll say about 10 years, maybe a little longer, we found out that my oldest son had RP. And once we figured that out, we began to reach out to specialists in the Austin area to try to figure out, okay, what is this? We had never heard of it before. So it's like, where do we go from here? So then we kind of started reaching out to other professionals in the Dallas area and started participating in these clinical trials, and then we had the gene testing and all that good stuff. And then later, we come to find out that not only did my oldest son have it, but my youngest son also have it. And then as time went on, through these trials and getting more testing, we found that my grandson has it, and also my granddaughter. So this is why we actually participate in these walks, in hopes that at some point, a cure will be found, even if it's on a minute level. Because of course it was very frightening at the beginning when they first found out that they had this disease. And I kind of heard it from some of the folks here, from talking to some of the people out over in the area over there, that it's a little bit scary at the beginning. But I say, just hang in there, stay positive, connect yourselves to the right folks, and, you know, try to ask the right questions. Connect yourself to other people who have the disease, perhaps, or other people who have any type of degenerative eye disease. And just, I think resource is how I'm pretty much going to put that. So that is why we participate in the walk. I think we've been participating like 8 or 9 years. Steph, you may have to fact check me on that. But it's probably been about that many years that we've actually been participating. And I'll tell you one thing, Michelle, I am super, super glad that we're actually having this in the Austin area. Thank you so much.
Thank you so much, Cece. And she is our fantastic Austin Walk Chair. So why do we walk? Number one, we raise much needed research funds to help us to continue spreading awareness and providing cures. And to bring the research and science to the public, these meetings included. Another reason why we walk is to raise awareness, let our community know who we are and why we matter. And lastly, to support our loved ones who are affected with a retinal degenerative disease. So if you have not registered for the walk already, you may ask, how can I get started? So you can register today. That's the first thing to do. In everyone's folders that they received, there is a guide on how to register. So that is a really nice resource. And next, I would recommend setting an ambitious fundraising goal. Also, it's really wonderful for everyone to share your personal story. Let people know how this is important to you and share why you are walking. Next, personalize your Vision Walk page. You can put a little description of yourself, why you're walking again, add a photo if you'd like, and send emails, letters, text messages, anything to spread the word that you're participating and to get other people involved through fundraising and registering them as well as part of your team. Also, social media is a wonderful aspect that we can utilize these days. We have Facebook, Twitter, YouTube videos. A lot of people these days use Facebook fundraisers as a fantastic tool to engage people to register or to fundraise for the Vision Walk. And then I always say it's important to ask 3 times. The first time, maybe people don't get back to you. The second time, some other people may get back, but not everyone. And the third time, hopefully by then you have responses from everyone. And the last thing that I would say is the most important is tell everyone you know that you are participating in the Vision Walk. And this is very important as well, how to get your community involved. So it starts with you in particular, and then surrounding yourself, you have your family, your neighbors, your friends, and your employer. And outside of that initial circle, there's the second circle of individuals that you connect with, such as your doctor, could be your retinal specialist, could be your regular primary care physician, your place of worship, a mechanic, a hair salon, a nail salon, a daycare, a dry cleaner, a civic group, and your school. And by being able to connect with all these different groups of people that you speak with on a daily basis or on a monthly basis and share your story and why you're walking, it's a great way to get people to donate to your event or to donate to your page. Become foundation ambassadors. I always like to say it's better to be safe than sorry and have information on hand because you never know who you're going to speak with. Keep your Vision Walk brochures and posters in your bag and hand them out to people. For example, if you go to a doctor's appointment, what better is it than to leave a few Vision Walk flyers at your doctor's office? And your eye care professionals, as I mentioned before, if you go in for an eye care visit, leave some materials with them as well. And lastly, at your local coffee shop, library, community boards, etc., it's always wonderful to be able to share and advertise the event, and specifically your team page if you're working to fundraise. Another wonderful tool that we always like to share with our members and constituents to utilize is letter writing and email campaigns. Now, I understand these days many people do not write letters and much prefer sending an email. But to make a personal letter and to share your story and why you are walking with your circle of people and ask them for their support and explain how to give is a wonderful way to encourage others to give to your team page. Another aspect to get people involved in your fundraising efforts is by creating some personalized fundraising events. This will allow you to boost your personal or team fundraising with a personal fundraising event. Now, there's many different ways one can do this. A few off the top of my head, there can be paint parties, wine tasting parties, jewelry parties, bake sales. Restaurants will often allow you to schedule a fundraiser or a certain percentage of the proceeds will go to you, and things of that nature. Another way for people to be able to give to the Austin San Antonio Vision Walk and any of our Vision Walks is through matching gifts. Many very large corporations will match their employees' charitable contributions, and I would just recommend reaching out to the Human Resources Department on how to get started. Some major companies that do this include Bank of America, Costco, Home Depot, FedEx, and Verizon. But I always will say it's better to ask and see what they have to say. Some companies will match up to a certain percentage, and it's just wonderful to speak with the Human Resources Department to get a better idea of how that works. We need you. As I mentioned, this is the first year that the Austin San Antonio Vision Walk will be located in Austin, and we are absolutely excited and honored to be able to have it in this wonderful city, and we would love to have you all participate. What to bring to the walk? So first and foremost, any extra donations you did not send in ahead of time, some people may have cash, checks, whatever, we are happy to take it on the day of the event. And your teammates in their team t-shirts and your Vision Walk banner. Not everyone does this, but many teams will create personalized t-shirts for the event and create a banner to walk with on the walk route. And lastly, your enthusiasm. The more enthusiasm you bring to the event, the more lively it is. I'm happy to introduce that we have a brand new website and platform that we are using for the Vision Walk that is more accessible. And this is an image of our new website that has a background picture of individuals from last year's San Antonio Walk. And in the forefront, it says Austin San Antonio Vision Walk, Saturday, May 2nd, 2020, 9 a.m. registration. And there are 2 buttons you can click, one to register and one to donate now. I would love to see you all on May 2nd. You can register today. The website is https://give.fightingblindness.org/Austin San Antonio Vision Walk. Now, I would like to discuss a little local involvement, and I'd like to introduce our chapter co-presidents, Todd and Meredith Dunn, to the stage. Thank you.
Hello, everybody. How's everybody doing? Glad to see everybody here. My name is Todd Dunn. This is my wife, Meredith Dunn. And as Michelle said, we're your chapter co-presidents for the Austin San Antonio chapter. And we're going to talk to you a little bit about some of the things that the chapter does and how we get involved. I think I changed your thing. Sorry about that. So first of all, I'd like to thank everybody here at the Travis Association for the Blind. Let's give them a round of applause for such a wonderful facility. First time I've been here. It's really, really nice, very impressive. All of our volunteers, the speakers, the exhibitors that are out there, and all the FFB staff for putting on and organizing this wonderful event today. I'm going to tell you a little bit about my story. I'm going to keep it short because I know you got a lot of questions, as we all do, for these excellent speakers with their knowledge of information. Around 2009, my son, Jameson, who's now 20 years old, he started having severe vision problems, and it started probably when he was about 6 or 7 years old. And I liken it to, I always tell the story about when he played baseball. He was a great little baseball player, catch everything, hit everything, did really, really well. And then the following year, he went from a really good player to somebody who couldn't even catch the ball, couldn't hit the ball, didn't know where first base was running the wrong direction, and didn't know what was going on. So we took him to several doctors, started seeing different eye doctors, and nobody could tell us what was wrong. They just said, put some glasses on him, he'll be fine. Put glasses on him. 6 months later, same problem, getting worse. Took him back, and of course we kept hearing the same excuse as maybe parents have heard, oh, he just doesn't want to wear glasses. He doesn't want to be seen in glasses. Nope, that's not the problem. So about 3 doctors, 4 doctors later, we finally found somebody who looked into his eyes and said, something's going on, this isn't right. And they finally diagnosed him with Stargardt's. And to hear that as a parent was shocking. They told us, he's gonna go blind, teach him Braille, he's probably gonna need a service dog. That's about how loud it was in the room after the doctor said that. It was terrible. But as we know, Stargardt's patients don't lose their vision, they keep their peripheral vision. And he's a junior in college, doing very well, straight A student, don't know where he got that from, but he's doing really good, and very proud of him. So at the time we learned about it, didn't know about the Foundation Fighting Blindness, never heard about it. We've never heard about it. Luckily, we were at a luncheon with some neighbors and just happened to talk about our kids, and their child, their son has a retinal disease as well, a different one, and started talking about the FFB. And so we started listening to him, and sounded exactly like what we needed, the resource we were looking for. So we researched it, got online. We weren't able to attend the first Vision Walk in San Antonio because we learned about his diagnosis afterwards. But we started going to the vision walks, starting with the second annual vision walk and been going to them ever since. And that was in 2009. 2012, the FFB came to us and asked us to be chapter co-presidents because the current president was moving on to another city. So we said, sure, we'll do it. So we've been chapter co-presidents ever since. That was in 2012. And some of the things that we do as chapter co-presidents, we help with phone calls. We have a phone call every third Tuesday of the month. Michelle sets these up, and we get on the phone call and we discuss chapter events and things that are going on to keep everybody informed. Anybody here wants to join those phone calls, you're welcome to. Get with Michelle, and you'll receive an email with the phone number and the access codes. You can join those phone calls. We also help organize socials. In San Antonio, we've had a bowling social, get together a bowling alley. We also had one at an ice cream shop where people can just get together and just sit and talk and share stories, get to know each other. This is a place where everybody has something in common, to not feel like we're not talking about fundraising, we're not talking about specific things, just sharing our own personal lives and getting to know each other. So some of the things that Michelle talked about the vision walk, but the different fundraisers. So some of you, first of all, let me ask, who here has never been to a chapter meeting or any phone call or anything, but just by show of hands, anybody who's brand new to the chapter, San Antonio Austin chapter? That's great. There's a lot of new faces here. I'm glad to see that. So some of you probably are learning, never heard about this before, never knew there was anything in San Antonio and Austin. So just like I was when we first learned about my son's disease. So when you go through the steps that Michelle talked about to set up your Vision Walk page, you may say, how am I going to raise money? And that's what I thought. How am I going to raise money? Where am I going to get this money to contribute to this wonderful thing? And we set a goal. I think the first year we set a goal of $1,000. We thought that's a pretty lofty goal. There's no way we're going to raise $1,000. If we do, fantastic. So I started sending out emails and telling our story, my story, to all my friends, coworkers, family, neighbors. Within days, we were well past $1,000. It was amazing, couldn't believe it. And this was our average donation was $50 or less. $50 or less was our average donation, and we went well past 1,000. So I raised it to 2,000. Within a couple of weeks, we were past that. The money just kept coming in by us just telling our story, sending out emails. So we raised it to $5,000. I thought, well, it'll be nice if we get there, but we probably won't. Month later, we were past $5,000. We ended up raising about $12,000 the first year just through mostly emails. And like I said, the average donation was $50 or less. So like you have to cast a wide net, like Michelle said, cast a wide net, ask everybody, talk to everybody you know. You can also go to places that you know, the places she mentioned, the restaurants and the diners and the businesses. And you can talk to them about setting up a fundraising evening, an event for dinner. Say, I'd like to set up something on this certain day. You're going to put the word out to everybody, and the business will donate 10%, 15%, or 20% of their proceeds towards your event, and that goes towards your fundraising goals. That's just an idea you guys can do. One thing we've done is we host a concert. We've done it many years in the past. At a friend of mine has a bar, and we'll host a concert at his venue. Get the band to come play. Some bands will play for free or very, very little money. And you get the word out. You don't really call it a cover charge at the door, but it's a donation at the door. So people will pay a donation at the door, and then the bar will also donate a part of the proceeds from the sales of that night towards our concert. And we usually raise about $2,000 just for that one night, just by getting the word out there. My sister would also host a silent auction at the concert. She would get items donated, set up a table like this, and people would come and bid on it, and she would raise money for her team. She raises money for my son. She doesn't have anybody affected in her family on her side. But so these are just ideas, or a happy hour. You could do something like that. Here's a great idea. My friend Brian Tully, who's down here, raise your hand, Brian. There you are. He's sitting at the front. He made these baseball caps. It says Foundation Fighting Blindness on it. It has the FFB colors, blue and yellow, and he is selling them right now. He's got a box of them. So this is a great idea here. I have some at home. If you want one, come talk to Brian. He is selling these as part of his fundraising goals. And again, email everybody, ask everybody. The donation you're not going to get is the one you don't ask for. That's all I can say about that. You need to ask, ask, ask. And it's amazing how generous people can be once you tell them your story. So the other thing is the retina practice outreach. And Michelle, do you want to talk about that? Do you want me to mention that briefly?
Hello. Okay, we're going to use this mic. So Retina Practice Outreach, and first of all, I would like to thank Todd and Meredith very much for being a part of the chapter and for speaking today. So I just want to give them a round of applause. Thank you very much. Retina Practice Outreach is an essential part of our chapter that allows members to volunteer their time to reach out to different retina practices in the region that we may or may not have formed relationships with in the past. And the purpose of this is twofold. One is to share our materials with them in regards to My Retina Tracker, as well as our genetic testing, as well as to provide them with materials for people who are newly diagnosed. And this way, people who are newly diagnosed have something when they go in and their doctor may diagnose them. So it's a wonderful way to get involved and volunteer within your local community. It could just be reaching out to the one retina practice where you personally go see your ophthalmologist, or your eye doctor where you go see your OD low vision specialist at. Both of these are wonderful ways to connect with someone in a practice in which you personally are involved in, and share what we do as a foundation and how our work can benefit the practice as well. So at this time, I want to thank you all for your time, and I hope you all join me at the 12th Annual Austin San Antonio Vision Walk. One little note, I want to just remind everyone to fill out the half sheets, as we will be having a raffle at the end of the day. And now we're going to take a 15-minute break, and there will be some snacks in the kitchen as well as some waters. Okay, everyone. So I would like to introduce our fantastic senior outreach, excuse me, I'm tripping on my words. Senior Director of Scientific Outreach and Community Engagement, Mr. Ben Shaberman. He has a wealth of knowledge, and he's worked for the foundation for over 15 years. So if we could all please give him a round of applause, and he will be giving us a research update on what is going on.
So thank you, everyone. It's a privilege and a pleasure to be here. Okay. I actually came to Austin for the first time right after Christmas for 4 days, just a little vacation, and I had a great time. I ate a lot of tacos. I went to Lady Bird Lake and ran back and forth. Isn't that more of a river than a lake? But anyway, and spent time on Rainy Street. So I kind of fell in love with Austin, and when I was asked to come back, I'm like, definitely. So I love it here. And thank you all for coming out and potentially supporting Vision Walk. Really, it's communities like Austin, San Antonio, and communities around the country that make what I'll be talking about possible. So you got a really nice introduction to inherited retinal diseases and clinical trials and some emerging therapies from the panel. And they stole a little bit of my thunder. So I might be repeating or going over some material that they covered. But I know for me, when I hear things 2 or 3 times, I learn them much better. So I'm hoping that's the case with you. I'll be talking about some various treatments, gene therapies, stem cell therapies, some other approaches, pharmaceuticals. But before I begin, I have a couple of introductory slides, and I'm also going to give you an introduction to the retina, which I think will be helpful if you're new to this world. So, as you've heard already today, the Foundation Fighting Blindness was founded almost 50 years ago by a small group of families who were affected by retinal diseases like retinitis pigmentosa. And they learned very quickly that virtually nothing was going on to even understand what RP was. And so they got together, they formed the foundation, and for the first really 40 years of our history, a lot of the research underway was to understand the retina, to understand how these diseases happen, to create models. A lot of mouse and rodent and even some canine models have been developed so we could test treatments. But those first 40 years were mainly lab research. There were virtually no clinical trials underway. But in maybe 2006, 2007, we began to move some of these promising treatments into human studies. And it's very exciting today that there are somewhere in the neighborhood of 38 clinical trials now underway. And virtually all of those were made possible by foundation funding. So that first 40 years was a real slog for us, but now it's starting to bear some very exciting fruit. And in my talk today, you're going to be hearing about a lot of different companies, Nightstar, AGTC, who you've already heard about, Spark Therapeutics. All of these companies were made possible, their existence was made possible by research that we funded. And that continues to be an important role for us, is to move promising therapies out of the labs and into clinical trials where companies are then excited about investing in them. Because companies often don't invest in treatments until they're proven in a lab study, or at least partially proven. So again, that continues to be an important role for us, is to get these therapies to the point where we can attract outside investments. And today, our portfolio of projects, we have about 80 projects. Many are in the US, but we fund a lot of research in all over Europe, in Israel, Mexico, China. We fund research, the best research. It doesn't matter where it is. We just want to fund the projects that have the most potential for restoring and saving vision. So that's a little introduction to where we are in this quest to overcome the entire spectrum of inherited retinal diseases, including age-related macular degeneration. So let's go through a little introduction to the retina. So when light comes into your eye, it goes through the outer covering, the cornea, the lens, then it goes through the middle of the eye, which is this gel-like substance called the vitreous, and then ultimately it hits the back of the eye, and that's where the retina is. And the retina is a very thin piece of tissue. It's only 0.5 millimeter thick, and it lines the back of the eye, much like wallpaper. Sometimes that's the analogy that we use. And what the retina does is when that light comes in, it absorbs that light and it converts it into electrical signals that are sent to the back of the brain, and your brain creates the images that we see. So the retina, as we often say, it's an old, tired analogy, but it works well. The retina is like film in a camera, or today's cameras, we have digital sensors. We don't use film all that much anymore. And so the retina is essential to making vision possible. It's that very special tissue that does just that. So this diagram up on the screen here on the left is a side view of the retina. So you're looking at that 0.5 millimeter piece of tissue blown up considerably. And I want to focus on a couple of different types of cells that are very important, especially in our discussion. And those longer vertical cells toward the top of the diagram are what we call photoreceptors, or the rods and cones. And these are the cells that actually convert light into electrical signals. Photoreceptors make vision possible. The cones are concentrated in the macular region, the center of the retina. Cones give us central vision. They give us our ability to read and drive and recognize faces. They give us color vision. They give us vision in a setting like this, where it's very bright. Rods, on the other hand, give us peripheral vision and vision in dark settings. And each of our retinas has 125 million photoreceptors. So there's an incredible amount of visual power that's packed into the retina. And then above the rods and cones is this single layer of cells, and it may be hard to see, but the photoreceptors are nestled into this single layer of cells. Those cells are called RPE cells, retinal pigment epithelial cells. And those cells provide support for photoreceptors. And in some diseases, the photoreceptors are affected first. In other conditions, the RPE cells are affected first. But if the RPE cells degenerate, because they're a support system for photoreceptors, then the photoreceptors suffer as well. And then to the right of this layered diagram, these different layers of cells, I just have a close-up image of a rod and then a cone. And you can think of them as like sensory cells, almost like very small hair cells that are able to absorb and respond to light. So as you've already heard today, inherited retinal diseases are caused by mutations in genes. And most of the conditions, there's just one gene that's implicated. And as you've also heard, there are somewhere in the neighborhood of 270 genes, each of which, when mutated, can cause a retinal disease like RP, Stargardt disease, Usher syndrome, and there are many other conditions. So what researchers thought of early on, and we began funding this approach somewhere in the neighborhood of 25 years ago, is that if we could replace that mutated gene with a healthy gene through gene replacement therapy, that might be a great way to halt retinal diseases. So what researchers began doing is they engineered viruses to be the delivery system for these therapeutic genes. And on this slide on the left, I have a hypothetical gene, piece of DNA, and it's being inserted into this colorful knobby ball that is actually the shell of a virus. We call it a capsid. So that ball is the delivery system, and it's carrying a gene hypothetically to photoreceptors or maybe RPE cells. And so in a gene therapy, you have millions of these capsids delivering millions of copies of the gene so that it's absorbed into the photoreceptors or other cells that are being targeted. And so I mentioned in the previous slide that there are about 38 clinical trials underway for emerging therapies. Well, about 25 of those are for gene therapies. Gene therapy is a very elegant way to treat retinal conditions. It's not the only way. It's not going to work for everybody, but it is a very effective way. And fortunately, it's been working relatively well so far. And really, our first big success, and this really made ophthalmological history back in 2 years ago, was Luxturna, which has been developed by Spark Therapeutics. I'm curious, before today, how many of you heard of Luxturna? If you can raise your hand. That's good. If you're in this world, everybody should know what Luxturna is. So this is a gene therapy that delivers RPE65 to replace the mutated gene, RPE65, in some people with LCA, labor congenital amaurosis, and RP. So it's just one of those 270 genes. But back in 2007, the first clinical trial for this approach began at Children's Hospital of Philadelphia. And early on in this trial, kids, young adults were having dramatic results. They were putting away their white canes. They had severe vision loss before treatment. They could see their parents' faces for the first time. Some were even seeing stars in the sky. So this was a very dramatic win early on in this clinical trial. And I'm very proud and happy to say that the Foundation Fighting Blindness funded about $10 million in research for RPE 65 that made that clinical trial possible. And so the clinical trial moved along. Again, it began at Children's Hospital of Philadelphia. And to raise more money to get the treatment across the finish line, Children's Hospital of Philadelphia spun off a company called Spark Therapeutics. And then in December of 2017, the treatment, it was named Luxturna, became the first FDA-approved gene therapy for the eye or any inherited condition. I think it was the second FDA-approved gene therapy for any condition that got regulatory approval in the US. So not only is this a great story for people who have RPE 65 mutations, it showed researchers, companies, and the rest of the world that gene replacement therapy was an effective way to treat these conditions. And so now you have a lot of other researchers and companies that are getting into this space. So this project really paved the way for that. So one company that got into this space a few years ago is called Nightstar Therapeutics. And they have a couple of clinical trials underway for inherited retinal diseases, one for choroideremia and one for X-linked retinitis pigmentosa. The choroideremia trial is now in phase 3. They've concluded their recruitment, so they've got all the patients they need. They had some impressive early results. If they continue to get good results in phase 3, then they would seek regulatory approval either from the FDA here and or the European Medicines Agency. Nightstar is a British company, so it's not clear where they would go first to get regulatory approval, but I'm sure they would want it in both Europe and the US. Interestingly, they were acquired by Biogen about a year ago. So the Foundation Fighting Blindness was also involved in the development of this treatment. We funded gentleman Miguel Seabra at the University College of London and Moorfields Eye Hospital for many years to understand the gene, create a mouse model of choroideremia, and then develop an early version of this gene therapy. Nightstar Therapeutics, again, also has a trial underway for X-linked retinitis pigmentosa. That trial isn't quite as far along. They've had some encouraging early results, and now they are in the process of moving this into a phase 2-3. So for those of you that heard Jill Dolgen from AGTC speak, they have clinical trials underway for X-linked RP and achromatopsia. And just to add on a little bit of what Jill said, they've had encouraging early results in both trials. For X-linked RP, there have been some modest but encouraging vision improvements for people that have had the central retina treated. And then for people who've had the peripheral retina treated, their vision has been stable, which is also an encouraging thing. So they continue to recruit for that, especially at the higher dose levels, and hopefully they can move that into a phase 3 before too long. Another condition which they're aggressively addressing is called achromatopsia. Achromatopsia is an interesting condition because it causes day blindness. So many of us here affected by something like RP, night blindness is the issue. You can't see in the dark. Well, imagine if you had a condition where you can't see when it's light. That's what achromatopsia is. And people with this condition have very, very strong sensitivity to light. So being in a room like this would be very uncomfortable and blinding. So in their achromatopsia trials, early on, they are seeing suggestions of improved light tolerance, which is a sign that the therapy may be working, still at an early stage of evaluation. They also had a clinical trial underway for a condition called retinoschisis, which is a splitting of the retina. The results from that were not all that encouraging. So that project, they're probably not going to continue development of that. And part of the reason I point this out, and this was made, the point I'm going to make was made with the panel discussion, is a lot of clinical trials don't succeed. And that's why we need to launch so many trials so that we can get a few of those therapies across the finish line. And just quickly, that's just a very small snapshot of some of the clinical trials for gene replacement therapy that are currently underway. There are others underway for choroideremia, Usher syndrome type 1B. Sanofi has a trial underway for Stargardt disease, and many other companies are trying to move therapies for Stargardt disease into clinical trials, including AGTC. There are more and more RP gene therapies underway for, in this case, for PDE6 beta and RLBP1, which was mentioned. And then an investigator that we funded a long time at the University of Florida, Shannon Boy, she has gotten approval to launch a gene therapy trial for LCA caused by mutations in the gene GUCY2D. So up to this point, we've been talking about gene replacement therapy, where you're replacing the entire gene, that entire gene that has a mutation, with a whole new healthy gene. Well, as you heard about in the panel discussion, another approach is to edit the mutation. So you're not replacing anything, you're going into the patient's retina and you're editing their existing gene. And the most popular way to do that right now is with CRISPR-Cas9. And in some ways, this is more elegant because you're just going after the mutated portion of the gene. The challenge is that there are literally thousands and thousands of different mutations. So because CRISPR is targeting one specific mutation, you would need a lot of CRISPR treatments to address those thousands and thousands of mutations. Fortunately, the CRISPR folks are getting smarter and finding ways to develop these editing tools that can address more than one mutation in maybe a region. So the first gene editing trial to get underway for retinal disease was launched by Editas and Allergan. That happened pretty recently. It's a phase 1/2 trial. They're currently recruiting for people who have a specific mutation in LCA. That mutation occurs in a gene called CEP290. And they're also working to advance CRISPR treatment into a clinical trial for a region of the USH2A gene called exon 13. And the foundation is funding about 5 other projects that are using CRISPR technology. Those aren't in clinical trials yet. The one that comes to my mind, because I've been talking to a few of our donors about it, is for a gene called RP1. And Mass Eye and Ear in Boston is working on that approach. So we've talked about gene replacement. We've talked about editing a gene. Now we're really going to get into the weeds here, but I think it's important to cover this. This is about the most technical that I'm going to get. So to really simplify things, your DNA is like the code in your cell. But there's a messaging system that's using that code to build proteins. And those messages are called RNA. Well, some very innovative companies are addressing the mutations in the message, in the RNA. And one of those companies that we're providing significant funding to is called ProQR in the Netherlands. We're investing about $7.5 million in their approach. So they launched a trial of this approach. It's called an antisense oligonucleotide, AON for short, for a CEP290 mutation. And this was the first RNA approach for a retinal disease. And what's really exciting is in the phase 1/2, about 60% of people in that trial had some vision improvements. And keep in mind, in a phase 1/2 trial what you're really going after is safety. You want to make sure it doesn't do any harm. You're hoping to see some efficacy, but often we don't. You have to move it into the next phase. So to see encouraging results in a phase 1/2 trial is a really good thing. So given the good results in the phase 1/2, ProQR has now moved this approach into a phase 2/3. And we're actually funding the advancement of the USH2A exon 13 treatment that's in an early stage clinical trial right now. It's in a phase 1/2. That began a few months ago, and they're recruiting for that. ProQR is also targeting a different mutation in USH2A, Stargardt disease, a mutation in the ABCA4 gene, and also another form of LCA, which they haven't publicly announced yet. Okay, so we've talked about replacement. We've talked about editing the DNA. We've talked about this RNA approach, masking the mutation in RNA. And now we're going to move to yet another gene therapy approach, which is really cool, and it's called optogenetics. So if you go back to that diagram of the retina I showed you, we talked about how you need photoreceptors to see. Those are the cells that make vision possible. And if you have advanced vision loss, if you're blind from RP or age-related macular degeneration or whatever condition, if you're completely blind, by definition, you've lost all your photoreceptors. Well, all these gene therapy techniques that I talked about aren't going to work for you because you have no photoreceptors left to treat or save. But alas, some very innovative researchers came up with a different approach to try to restore vision in people who have this advanced situation. And what they realized or observed is that these other cells in the retina survive after the photoreceptors are gone. And these are called ganglion cells. And so I have a big ominous X through the photoreceptors in this diagram to signify somebody who's lost all their vision. But then I have a more hopeful oval around the ganglion cells that may still be harnessed for vision. And what the researchers have done is they've delivered a gene that makes these ganglion cells light sensitive. So in very simple terms, it enables those ganglion cells to operate somewhat, and I put that word somewhat in air quotes, like photoreceptors. And what's really cool is this approach really got on our radar screen, I want to say maybe 8 years ago, and now it's in clinical trials. Allergan and GenSight both have this in a clinical trial. AGTC is also partnering with a company to move this into a clinical trial as well. So the real advantage here is you're offering the potential to restore vision to somebody with the most advanced disease, and it doesn't matter what mutated gene caused their disease, their vision loss. This is agnostic to the mutated gene. Now, the cautionary note is we still haven't gotten reports from the clinical trials. We're still not sure exactly what patients might be seeing. It's still at an early stage. And for some of these approaches, there are glasses or goggles that are included to help enhance the information that's coming into these ganglion cells, to enhance and amplify that visual information. So stay tuned. Will these be a home run right out of the gate? Well, we hope so, but even if not, we will learn a lot from these early trials so that we can do it better a second time. And actually, the foundation is looking at funding even better approaches to optogenetics that hopefully you'll be hearing about in the not too distant future. So that's just a quick, or maybe not so quick, overview of gene therapies. And I want to talk a little bit about stem cells. And you heard some good information about stem cell therapies from the earlier panel. So the real benefit of stem cells is you can use them to either replace cells that have been lost, or you can use them to support or keep healthy existing retinal cells. So there's the replacement paradigm where you're replacing the entire cell, where you're trying to save the existing cell. And what's really cool is today researchers can take a patient or any person's blood or skin and genetically tweak those cells so they revert back to a stem cell state and coax them forward to become retinas. So stem cells, by definition, have not matured into bone or blood or retina. So if you can convert skin or blood to become a stem cell, then you can make it into anything you want. And researchers are now doing that. We fund a guy named David Gam at University of Wisconsin-Madison, who has a company, Opsis, that was formed a few years ago, who's moving stem cell therapies using this approach into clinical trials. He's not quite there yet. But I wanted to give you an overview of a couple trials that have been underway. One is using a stem cell that is almost matured into photoreceptors. And the paradigm for this approach is neuroprotection. The idea is to save or preserve what somebody already has. It's targeted for people with RP, Usher syndrome, and some of the other inherited conditions. The company is called J-Site. They launched their trial a few years ago. In the phase 1, they had some encouraging results at the highest dose. The treated eye in the patients performed a little better than the untreated eye. So they moved it into a phase 2B. Now they're looking at it in 85 patients. They haven't reported anything yet. Hopefully we'll be hearing about this approach in the not too distant future. And then a similar cell type has been developed by a company called Reneuron, and this is being evaluated at Mass Eye and Ear in Boston. But the difference is, instead of trying to protect or save, you're actually trying to replace retina that's been lost. And so this trial has been launched at, again, Mass Eye and Ear in Boston, and they also have a site in Phoenix at a private practice there. And in the second phase of this study, the early results are showing that the treated eye, they've been able to improve vision by a few lines for many of the participants. So these people aren't completely blind. They have a little vision, but by putting these cells in, they're bumping up the vision by a few lines. These results are early. They've only been observing these patients for a few months. We really need to see how long this effect is sustained before we can get too excited. But again, this is for people with RP, Usher syndrome, and potentially other conditions. The foundation has been funding work for this project for a long time that made the clinical trial possible. And then finally, in people with age-related macular degeneration and Stargardt disease, if you remember those RPE cells I was talking about, those are the cells that are affected first in those conditions, those macular conditions. And there are a couple companies, there's actually a few companies that are working to use stem cells to build new RPE to replace the lost cells, so you can save photoreceptors. And one company is out of Southern California called Regenerative Patch Technologies. They launched a clinical trial. Now it's been a year and a half, 2 years, for people with dry age-related macular degeneration. And what they're doing is they're putting new RPE cells derived from stem cells on a scaffolding so they're better oriented in the retina when they're transplanted, and the scaffolding actually helps these cells survive better as well. So in their early results, they showed some promising activity for the first few patients, but it's still, again, very early. And actually the researcher in this study who's developed the scaffold, Dennis Clegg, is collaborating with that other researcher that I mentioned early on, David Gam, when I began talking about stem cells. So generally speaking, stem cells are not quite as far along the development path as the gene therapies, but there's some good progress being made. But a key advantage of most stem cell therapies is they're designed to work independent of the mutated gene. So keep that in mind. I wanted to move along and talk about some other therapies, pharmaceutical therapies that are in clinical trials. And one is a very powerful antioxidant being developed by a company in Dallas that we're funding. We're providing up to $7.5 million to this company. They're called Nacuity. In retinas that are affected by genetic diseases and age-related macular degeneration, oxidative stress is very high, and oxidative stress exacerbates the underlying retinal condition. Well, this company, Nacuity, has developed a molecule they called NACA, which in animal models showed some excellent results in terms of mitigating or tempering oxidative stress to try to save vision. What's nice about this approach is it's oral. It's something you take orally, so you can modify the dose. You can try a lower dose. You can try a higher dose. If it's not working, or if for some reason there's a safety issue, you just stop taking it. Whereas with a gene therapy or stem cell therapy, once you get those, it's hard to stop those or reverse those. So this is actually in the clinic in Australia right now. We're hoping the clinical trial for this begins in the US within about the next year. And then finally, there's a company in France called Sparing Vision, and they've developed what ultimately is a gene therapy, but this gene therapy simply works to keep cones alive, and it works independent of the mutated gene. For those of you who have RP or have a loved one with RP, you know that generally speaking, your rods degenerate first, and then later in the disease, the cones go. And what these researchers in France discovered is that rods produce a protein that helps keep cones alive. So they developed a gene therapy that expresses this protein, and the protein is called, very aptly, rod-derived cone viability factor, or RDCVF. So this is a very elegant way to try to preserve cones and to do so independent of the underlying gene mutation. And we've been funding this work for many, many years. We're currently investing 7 million euros to get it into a clinical trial, and that should begin within the next year. And then quickly, because I know I'm starting to run a little short on time, I wanted to mention that there are several therapies underway for Stargardt disease. Stargardt disease affects those RPE cells that I was talking about. They're basically, what happens in Stargardt disease is waste collects in the RPE cells. So what these therapies that I have on the slide from Alkeus, Acucella, and B-Light attempt to do is to slow down the accumulation of those waste products. And Alkeus, Acucella, B-Light, all their treatments are actually in clinical trials right now. Nobody has reported results yet, or results of efficacy, so stay tuned. There's another company called Iveric Bio that is attacking a different part of the retina, the retinal system, to try to slow down Stargardt disease, and that's actually part of the immune system. And that is in a phase 2. They haven't reported results for that yet either. So that's a really broad brushstroke of many of the therapies that are in clinical trials or soon to be in clinical trials, and there's a lot more going on. Like I said, we're funding 80 different projects. Most of those I haven't even talked about in this presentation. But I wanted to wrap up things by talking about genetic testing, and we heard a bit about that with our esteemed panel. So if you have an inherited retinal disease, finding the gene mutation is important for many reasons. First of all, it confirms your diagnosis if you can find the gene. Many retinal diseases can look alike. RP and choroideremia can be confused. In some cases, I've heard even Stargardt disease and XLRP can be confused. But once you find that mutated gene, then you know the underlying cause. With that knowledge, you have a more definitive diagnosis. You know who else in your family is at risk because you then know the inheritance pattern. And what's most exciting is knowing your mutated gene can help you qualify for a clinical trial. So there are a lot of great reasons to get genetically tested. And we offer, as you heard, a no-cost genetic test that any eye care professional, ophthalmologist, retina doctor, even an optometrist, can go to blueprintgenetics.com and order the test. What's important is the doctor goes to Blueprint. You don't go to Blueprint. They don't want to talk to patients. They want to talk to doctors. You go to your doctor and ask him or her to order the test. And they do this all online. They don't have to talk to the foundation. And as long as they can clinically diagnose you with a retinal disease, an inherited retinal disease, then you can get the test for free. The only exception is if you've been tested by a broad panel in the last 3 years, you have to wait for 3 years to occur before you can get retested. There are some really strong advantages of the panel from Blueprint. It tests a lot of genes, even a few genes outside of our scope. It tests 285 genes. It also includes the gene for X-linked RP, which other panels don't test. It tests for certain mutations called copy number variants and other non-coding mutations. I'm not going to get into what those are. But again, other panels may not test for those. And perhaps the most important thing to know about the Blueprint panel is they do not give away personal information to doctors or other companies when you sign up. Other panels may do that. So if you're considering other panels, obviously we strongly encourage you to use the one that we're participating with. But if you're concerned about your privacy, make sure you understand what's happening with your personal information. As part of this program, when you're tested, you get free genetic counseling, whether it's from a genetic counselor associated with your doctor or whether it's through Informed DNA, which is a company that we work with to provide genetic counseling. And they're excellent. There's no other organization in the country that understands the retinal disease space better than us. And they will help you understand what the results mean for you, your family, and they'll help get you on the path to managing your condition. So again, ask your doctor to order the test if you're interested. And then finally, even before you get genetically tested, we're hoping that you register in our patient registry called My Retina Tracker. Now, what we have to keep in mind is with inherited retinal disease, you guys are rare, even though there's a bunch of you in this room today. There are only about 200,000 people in the US who have these conditions. There isn't a lot of human data available to the research community about these conditions. So we want you folks to upload, to register your information on My Retina Tracker to make this information available to researchers and companies. They never get your personal information. They only get your disease information. If you know your gene mutation, great, you can put that in. If you don't know your gene mutation, that's okay as well. Your disease information is still valuable. So once you're in My Retina Tracker, you're helping researchers understand these conditions better, and you may get also recruited for clinical trials. That's another important benefit of the registry, is researchers are using it to recruit people for clinical trials. We have a total of 25,000 people who are registered, about 15,000 are active. We imported some old records that still need to be updated, so that's why there's two different numbers. And as part of this program, we've had more than 7,000 people genetically screened. But you don't have to be in My Retina Tracker to get the genetic testing. They're two different things. We really want you to be in My Retina Tracker, but you don't have to be. And it's been designed to be easy to use, to work with screen readers like JAWS, and to be as accessible as possible to people with low vision. And you go to it at myretinatracker.org. It's highly secure, it's free. You can go in and fill out maybe half the record and come back. You don't have to fill it out all at one time. So by getting into My Retina Tracker, you're getting on the radar screen of the research community and therapy developers. So we strongly encourage you to do that. So to close out my talk today, I have a little bit of shameless self-promotion. So I've been with the foundation for 15 years. And part of my job, I don't do it so much anymore, is I interviewed a lot of families and kids to do stories, human interest stories. And over the years, I've been tremendously inspired by the courage and vigor with which these families live their lives, especially the kids. So I wrote a sci-fi young adult novel, appropriate for anyone over, I'd say, the age of 12, about a kid who's born without retinas. And researchers are mystified. They've never seen anybody born without retinas. So a little bit of spoiler alert here. It turns out that he's actually an alien from another planet who's been swapped for a human baby at birth. And so the novel follows this alien on Earth. Nobody knows he's an alien. He doesn't even know he's an alien. As he grows up, and he grows up to have a really nice life, and he plays awesome lead guitar in a band and so on. And then the human baby is brought back to this alien planet of blind inhabitants. And ultimately, Retina Boy, who's the alien boy without retinas, he's called upon to save the Earth with his girlfriend. So that's sort of the plot. It's a lot of fun. I cover this space a little bit, but I really wanted to pay homage to the kids who live with these conditions so bravely and courageously. So it was published by Loyola University of Maryland. It's available on Amazon, on Audible. Of course, I had to have an audio version, and so you can order it there. I brought 4 copies today, and if you make at least a $20 donation to the foundation, and you don't have to do that now, if you want to give us cash, whatever, but we trust you that you will do this. You can go online and make the donation. I'll give you a copy, and I'll be happy to sign it as well. But again, it's available on Audible and Amazon. And if you want to learn more about it, it's at retinaboy.com. And I just have a new animated trailer, which I don't have here today on my website, 45 seconds. It's very cool. So that concludes my talking, and hopefully we have a little time for Q&A.
Sure. Let's give Ben Shaverman a round of applause, shall we? And if you do get Retina Boys signed, that will at least double the value of the book. So, you know.
It's an investment.
We're going to take questions one at a time, so please raise your hand and we will walk over to you. I'm going to start right here in the middle.
So quick question relating to the retina. So if you happen to have a retina precaution, if your retina happens to get detached for some reason, would that increase any kind of retina disease?
So are you saying you've had an injury?
No, I'm saying I do have a retina precaution, but if for some reason my retina gets detached.
So you're saying somebody has a retinal disease like RP. Are you asking, is it more susceptible to detachment, or are things going to get worse with detachment? I'm still talking to her.
I'm asking, would it get worse with detachment?
Oh, okay. So I wish the doctors were still here to talk more about that. A lot of it may depend on how severe the detachment is and where the detachment is. Because if it's further out on the periphery of your retina, it may not be such a big deal. If it's detached more toward the center, that could affect your vision more. That much I'll say. Good question.
Hi, I have 2 questions regarding genetic testing. One is, if a person has been tested a few years ago and like one of the presenters said, it may show no results, should that person go through another testing to see if results will show up? And then question 2 is if a family member's been tested and there's a potential another family member that needs to be tested, should they be tested under the same testing company?
Right. So those are good questions. So the retesting question is why genetic counseling is so important. A genetic counselor would help you answer that question. It's unfortunately a complicated situation. Everybody's case is different. It depends how long ago, it depends on the panel, and it may depend on what they've found. And a genetic counselor is going to help you understand whether or not retesting makes sense. With our program, if you've been tested with a broad panel within the last 3 years, we're not going to provide it at no cost. You would have to wait a few years. Now, in terms of getting other family members tested, if the other family member is showing signs of the condition, they should be able to get that for free. If you're just trying to see if they're a carrier or they may be affected but they don't show signs, then you can get family tested, the family member tested at a very small cost. It's like $99. Feedback. Okay, is that better? So, but again, I would strongly encourage you to talk to a company like Informed DNA, send them your report. They do charge for the counseling. About half the time it's covered by insurance, but they're going to help you understand what the next step for you should be.
What is the difference in getting genetically tested through Spark versus the blueprint that you're talking about?
Are the Spark people still here? No. So today, the Spark panel is about 248 genes. The blueprint panel is 285. The blueprint panel also tests for the gene X-linked that causes X-linked RP when it's mutated, RPGR. That's a pretty common gene. And then there are other types of mutations that blueprint tests for. Those are the non-coding variants and the copy number variants that Invitae and Spark do not test for. And then, as I mentioned earlier, talk to them about privacy. If you're concerned about your personal information being shared, we protect that. I would talk to them about whether or not they protect your privacy. Now, with that said, they've been at this, at least more broadly, for a little longer than we have in terms of the big market penetration. So more doctors may be using them now, but our panel, as I've been saying, is more broad and it goes deeper. Two more questions, I've been told.
Hi. With all this DNA testing going on, are they noticing any patterns in terms of certain cultures or certain regions in the world that are higher percentages of RP or Stargard's? Have they noticed any places like that?
If there is a Rafael Servino in the room, your access is here to pick you up. Just wanted to let you know. Okay. Sorry. Thank you.
So that's a great question. And for many years, we've known that certain genes run in certain ethnic populations. That's been known for a long time. And people can be carriers for these gene mutations and have no idea. And so when two people decide to have a child, you have no way of really knowing if that other person is a carrier. Really, like prenatal testing doesn't occur for these conditions. It really doesn't occur that much because they're not that common. But yes, there are certain genes that run in certain populations. For example, there are certain genes that are more prevalent in Ashkenazi Jewish people and also other people in the Middle East. And we funded Hadassah in Israel to screen as many people in Israel as possible. That included people with Jewish ancestry, Bedouins, Christian Arabs. I'm forgetting all the different ethnic groups. And that's a great way to learn about which genes are more prevalent in these populations. But French Canadians, there are some more common genes, and other populations as well. Guess we have one more question.
Over here. Hi, thank you. Yes, I understand that there is a specific microchip that is being implanted in certain patients made out of silicone that helps with photoreceptors, and it communicates with the brain to make you could say an image or see better with people with RP. How is that being done right now?
Sure. So the commercially available version of that is called the Argus 2, and that was actually FDA approved back in 2013. And the way that works is, this is for people who have no vision left. And they wear a pair of glasses with a video camera, and that video camera sends signals to the microchip that's implanted adjacent to whatever remaining retina is there. So the images are sent to that microchip, and then the microchip sends signals to the back of the brain. Now, it really doesn't provide vision as we think of it, colors, details, and so on. It provides some patterns to help people better live in the world, to better orient themselves. They can see movement and maybe shadows and shapes. And for some people, that's really a big deal if you have absolutely no vision. So that's the Argus 2 that was developed by Second Sight. There are other companies that are working on similar technologies that are hopefully more powerful. There's a company called Pixium out of France that has a clinical trial for their similar technology. It's a more advanced version, and they're hoping that provides even better vision. They're still at a relatively early stage in their clinical trial.
All right, let's all give Ben Shaberman another round of applause.
And then if anyone didn't get around to being able to ask Ben a question, please find him after the program is ended, and he will do his best to speak with you. And he has his books back there as well. So thank you everyone very much for joining us today for the Austin Vision Seminar. I wanted to take some time and thank our panelists, Mr. Ben Shaberman, and all of our exhibitors for coming today, and most importantly, all of you for joining us. And to Donna, I wanted to remind everyone to please complete your evaluation forms, and if you wouldn't mind dropping them off on the registration tables where you came in, that would be wonderful. We always appreciate feedback. And then lastly, in the exciting part, well, twofold. First is the exhibitor booths will be open once we finish our presentation until 1:30 p.m. There are still many out there if you have a chance to go take a look. And we will be doing a raffle. We have 4 prizes from the foundation and 2 prizes from 2 exhibitors that I will share as soon as they are picked. So first, we are going to do the exhibitor raffles, and he will be...
I am going to now reach into my magic bag of evaluation forms or raffle cards.
It's sticky, sorry.
And Ms. Goldfarb Shapiro will be announcing our first winner.
Thank you, Connor. I apologize if I mispronounce your name. I believe it's
Tarani Moragan.
You received a $25 Amazon gift card courtesy of the Tap V exhibitor. That's incredibly sweet. Thank you so much.
Unprecedented.
Thank you very much. Yes, we're going to pull another card.
Another winner.
From Tap V. They are an organization at the Texas School for the Blind and Visually Impaired for Children. The winner is Ellen Boisyan. Wonderful. Donna will bring you the gift card right now. She's in the purple shirt. She's in the purple blouse. Next, we are going to be giving a little package gift that has been given to us courtesy, or that will be given to you courtesy of Open Eyes Life Coaching and Consulting. It includes a Walgreens gift card, stress ball, as well as some hand lotion for all this dry weather. And their booth is still open in the exhibitor area. The winner is Christine Rogers. If you are here, please raise your hand. Thank you. Now, for the last 4 raffles, my apologies. Everyone will be receiving a Starbucks gift card courtesy of the foundation. And the first winner is, thank you, sir, Norberto Cardenas. He has his phone up. He's in the 4th row on the right-hand side. Thank you. Patricia Jackson. She is raising her hand in the back. You also receive a Starbucks gift card. They're connected, so you have to disconnect those two. And we have 2 more Starbucks raffles that we're working on right now. Miriam Morel. Can you please raise your hand? She's right there. And one last one.
And the winner is
Me.
It's me.
Thank you. Melissa McFarlane. Thank you, everyone, for joining us today. Don't remember to register for the Vision Walk if you haven't already. And if you're interested in buying My Retina Boy, Ben is in the back left corner. Thank you, and I hope you have a wonderful afternoon.